My name is Brittany.
And my name's Brittany.
And for our enzyme project, we chose DNA Ligase I.
So, to go over some background on DNA Ligase I, it was discovered in 1967 by Robert Lehman and laboratories. During the 1960s, other labs were looking at it, such as researchers Delbert, Richardson, and Hewitt's labs, but it was really Robert Lehman in 1967 that got the recognition for looking at the function and the mechanism of DNA Ligase I. Its enzymatic classification is EC6511. The 6 in the number scheme stands for what class it's in, and that is ligases, so hence its name DNA Ligase I. Its scientific name is polydeoxyribonucleotide synthase ATP1, but we'll stick to DNA Ligase I and L1G1 for the rest of the presentation. All living cells have DNA Ligase I, but there is a split between the cells, eukaryotic and prokaryotic. Eukaryotic use ATP as a catalytic substrate for the enzyme, while prokaryotic cells use NAD+ as a catalytic ability. So because we'll be talking about humans and mice and other eukaryotic beings, we're going to be talking about the ones that use ATP as its co-substrate. And also within humans, we do have a L1G1 gene, and the next slide we'll talk about what chromosome is on and where that's located. Finally, for DNA Ligase I, its functions are to catalyze the formation of the phosphodiester bond between the 3' hydroxyl and the 5' phosphate group. It seals the nicks of DNA during replication and repair. And finally, it joins the Okazaki fragments of the lagging strand. So some bioinformatics of DNA Ligase I. It's found on chromosome 19 and is denotated as the L1G1 gene. It's found in this area on the chromosome. It is made up of 919 amino acids and is 101,736 daltons. It's made up of 3 domains: NTACE, OB fold, and DVD. Just some background information on what these domains do. NTACE and OB fold are the minimal catalytic unit of this enzyme. They are conserved in all known DNA and RNA ligases. Their main job is to contain the active sites in which DNA will bind. And so, for instance, they have phenylalanine residues in which will bind to the minor groove of DNA, and those bonds will be stabilized by salt bridges made using residues of aspartate and arginine. The DVD domain's main job is to give DNA its binding affinity. It binds with DNA upstream and downstream of the nick made during DNA replication and repair. And then it also is the domain that stabilizes DNA in its unbound form. All of these domains are connected and touching each other, so they make somewhat of a ring-like structure around the DNA during replication and repair. So for this enzyme, they need cofactors and co-substrates to do its job correctly. So the cofactor for this enzyme is magnesium 2+, and the co-substrate is ATP. We'll go over kind of what their jobs are and how they help the enzyme do its job in the body. And also the predicted PI for this enzyme is 5.5.
So after the enzyme is produced, certain post-translational modifications have to take place. And on this line here, each of these tick marks represents one modified residue. So the first one we're going to talk about is lysine, and that's only modified one time, and it's acetylated, and that happens closer to the C terminal of the enzyme. And then the other two are threonine and serine. Threonine is phosphorylated a couple times, but serine is the most common, and it is definitely phosphorylated. And the cyclin binding motif of the OB fold domain plays a pretty big role in this phosphorylation of serine, and one of the most notable is serine 66, which is responsible for the interaction between replication factories and proliferating cell nuclear antigen, or pCNA, which we'll talk about in a lot more detail later. So here's the mechanism of DNA Ligase I, and the reaction always begins with the hydrolysis of ATP to AMP, which is covalently bounded to this lysine residue near the N-terminal and the active site, and then after that happens, the adenylated enzyme transfers that AMP group to the phosphate, the 5' phosphate on the DNA backbone, and then the nick is sealed when this 3' OH attacks that phosphate, and it creates a phosphodiester bond. And there isn't really any conformational change depicted here, but there is a 20-degree pivot of the DNA binding domain during this reaction.
So as I mentioned earlier, there are cofactors and co-substrates that enable DNA ligase to do its job. First, we're going to talk about the cofactor magnesium 2+. What it does is, it's shown in this picture, the two purple globs are supposed to denotate magnesium. They're two metal ions, and they bind within the active site of DNA ligase. And what their main job is to do is to stabilize the negative charge on the phosphate of AMP leaving group. And secondly, it's supposed to activate the nucleophilic attack of 3' hydroxyl onto the 5' phosphate to form the phosphodiester bond. Now the co-substrate of DNA ligase I is ATP, and it's kind of shown as the little blue part here in this diagram. And so what ATP's job as a co-substrate for DNA ligase I, it is supposed to give the catalytic ability of this enzyme to make that phosphodiester bond. It specifically binds to lysine in the enzyme domain, and the enzyme domain is responsible for holding the AMP cofactor deep within the pocket. DNA ligase I does not have residues available in which to create this catalytic ability, so that's where ATP comes into play. So we talked about how DNA ligase I functions and its mechanism. So now what we're going to talk about is when DNA ligase I malfunctions. One deficiency syndrome that we found with DNA ligase I deficiency, within these individuals there is immunodeficiency and an increase in sensitivity to DNA damaging agents such as UV radiation and X-rays. With these individuals, they have delayed growth and they show a poor proliferation of their lymphocytes, which kind of ties back to the immunodeficiency. This deficiency is very rare. It was only found in one case of one human, but this was replicated in rats, and so that's where they saw other parts of the symptoms. But again, it's very rare, but it is a devastating deficiency. Bloom syndrome is another malfunction of DNA ligase I. With these individuals, it is an autosomal recessive disease or disorder, but it has these individuals have high chromosomal breakages, and they have a predisposition to cancer, and they have genomic instability, which kind of ties back to those increasing chromosomal breakages. This is a very rare disorder, but it is also very debilitating. With DNA ligase I malfunction, there's an increase in cancer and neurodegeneration. And one other thing we found that was really interesting was if glutamate 566 residue is replaced by a lysine, individuals will have severe immunodeficiency and delayed processing of the Okazaki fragments.
So for our experiment, we found a study that was done in the early 2000s, and it was published in the Journal of Biological Chemistry. And what it dealt with was the functional complex that forms between DNA ligase and proliferating cell nuclear antigen. So there are a lot of facets to this study, and they're all listed here, but we're mainly going to focus on the three that we found the most interesting. And this is what pCNA looks like. It has 3 subunits, and they're all the same, so it's called a homotrimer. And what it does is, if this is your DNA, this is pCNA, it clamps around that DNA, and that stimulates DNA ligase to come in and bind to the DNA as well.
So again, we're going to go over only 3 experiments that we found were really interesting within this paper. So what I'm going to do first is just go over the methods that they followed, just kind of watered down in an overview. They used 11 oligonucleotide substrates, and they did 2 main assays, an enzyme assay and electrophoretic mobility shift assay. In the enzyme assay, they performed the reaction in a reaction buffer, incubated around 37 degrees, dyed with a formamide dye and heated it for 95 degrees for 5 minutes. They separated it on a polyacrylamide and urea denaturing gel, and they studied the products using a phosphodimer analysis. And then they also used the electrophoretic mobility shift assay. They completed the reaction in a binding buffer, incubated around 4 degrees Celsius for about 5 minutes, and then they separated it on a aggregose and polyacrylamide gel, and they looked at the products using a phosphodimer analysis. Now, the first experiment that we're going to go over and we found was interesting was how PCNA stimulates DNA ligase 1 activity. So first, looking at this gel A, is we have in the different lanes, lane 1 is just PCNA by itself, lane 2 is L1G1 by itself, and then 3 through 7 show an increase in PCNA concentration and how that acts upon DNA ligase and how much complex and product it makes. So as you see, and as the researchers saw, as PCNA concentration increased, so did DNA ligase 1 product increase. So that's what they were kind of hoping for. It shows that PCNA is increasing and stimulating DNA ligase 1 activity. Now, what is going on in gel B, PCNA works with a lot of different enzymes, and the researchers knew that. What they wanted to ask was, well, PCNA works so well with DNA ligase 1, does it have the same effect on just any old ligases? So what they did in gel B is they took another ligase, noted as T4 ligase, and they did the same exact thing. Lane 1 had PCNA, lane 2 T4 ligase by itself, and then 3 through 7 shows an increase in PCNA concentration acting upon T4 ligase. If you just compare the product line here compared to here, you see that PCNA really does stimulate DNA ligase more than just any old ligase, and that's what they saw, that PCNA does have this specificity for this enzyme DNA ligase 1. The next experiment that we found was interesting in this paper was how when they looked at PCNA enhancing the binding of DNA ligase 1 to DNA substrate. So just to break down this gel for you, in lane 1 was just DNA substrate by itself. Lane 2 was PCNA by itself. Lane 3 was the DNA ligase 1 by itself. And then 4 through 6 was all 3 substrates together. So PCNA, the DNA substrate, and DNA ligase 1. And with it, PCNA's concentration was increasing. As you can kind of see with this product bar, you see that as concentration of PCNA increased, so did the amount of products created. What's going on with lane 7 here is this is just DNA ligase 1 by itself. And what the researchers were trying to show was, yes, at high concentrations, DNA ligase 1 can create a lot of DNA complexes, but naturally within our body, we do not have these high concentrations of DNA ligase 1. So that's why it's necessary for our body to use PCNA to be able to create high concentrations of these DNA complexes we need.
So the researchers also studied the role of phosphorylation on DNA ligase because it's a substrate for casein kinase 2 phosphorylation. And 2 of these sites are serine 66 and serine 141 because they have optimal properties for casein kinase 2. Now, it's important to note that the phosphorylation doesn't necessarily inactivate DNA ligase, but it does prevent the interaction with replication apparatus. So naturally, the researchers wanted to find out if the phosphorylation had any effect on PCNA stimulation of ligation activity. So you can see here that there is no casein kinase 2, so there's no phosphorylation. And here there is casein kinase 2, so there is phosphorylation. And you can see here that there's not really much stimulation, but here as you increase PCNA concentration, you have an enhanced formation of product, which this means that PCNA does eliminate the stimulation of DNA ligase. So we have a couple practical applications, and the first is peptide antigen sequences. And here's one of 15 amino acids for DNA ligase, and 4 of them are negatively charged, 2 of them are positively charged, and this makes the isoelectric point about 4. And ideally, you want this sequence to correspond to a region that's exposed when the enzyme is in its native conformation. So hydrophobicity is one thing that's pretty important with this, because if you have a hydrophilic sequence, then that's more likely to correspond to one of these exposed regions. But if it's hydrophobic, that's more likely to correspond to one of the more internal regions, which is harder for the sequence to reach. And then second, we found a study that found the presence of DNA ligase is higher in tumor cells than in normal tissues. And they also found that DNA ligase antisense oligonucleotides suppress cell proliferation. So this suggests that DNA ligase plays more of a role in proliferating cells than in resting cells. And basically this warrants the testing and design of human DNA ligase inhibitors as new anti-cancer agents. And then lastly, we have genetic engineering, and it's pretty well known DNA ligase is used in genetic engineering. So for instance, in recombinant enzymes, after you use restriction enzymes, you can glue the sticky ends back together.
And here are references. Thank you for watching.